Or we'll continue on. Help me out here. Is this where I left off? Do you have notes on simple columnar epithelial tissue?
Yeah, where it has simple boil.
Okay. Yes, so we are in the right place. So we're continuing on with the epithelial tissues. And this is the next one. Let me turn my mic down a little bit. Test. Okay. Simple columnar epithelial tissue, ET. That's what it looks like. You even see some surface modifications there. I see goblet cell and microvilli as things I would note. Remember, the function of those is for microvilli to increase the absorptive area. But not of the whole cell. Look where it is. It's on the top. So it increases the absorptive area on the apical side. And the goblet cell function is to secrete mucus. So this is a tissue that is moist. And they have a bunch of locations there. It's a very common tissue. Let me give you a couple of locations. Small intestine, oviduct. Okay, just to name a couple. And the function of a tissue that has simple columnar cells is secretion absorption. Okay. Function. I have a picture of this tissue on the next slide, and what you can see, let's see here. Let me turn the lights off for this one, it's kind of hard to view. Let me get my pointer. The basement membrane is all folded in, okay? And the simple columnar epithelium, it kind of lines the outer edge, okay? This one, I see some cilia. It looks kind of fuzzy. The location of this slide, you should know, is the oviduct. The oviduct's function is to receive an ovulating egg once a month in a woman's, ovarian cycle, which you'll learn about next semester. And there's a lot of little nooks and crannies for the egg to wait for sperm to fertilize it. So this is kind of the environment it awaits. And so I would add one more thing. That is, sometimes you see cilia on this tissue type. The oviduct may be ciliated. All right, I'm going to move on. Simple columnar ET. Another type of columnar ET is one that appears stratified. If you look at the picture, I look at the nuclei. The nuclei are always your clue, okay? And this clue, it looks like it's stratified because you see nuclei stacked on top of one another, but what you may not know is that every single cell is touching the basement membrane. So this is our ciliated, pseudostratified columnar epithelial tissue. ciliated pseudostratified columnar epithelial tissue, ET, and you don't see goblet cells on this picture, but I usually do see it on the slides I showed you. So I'm going to tag on with goblet cells. In terms of locations, an example I'll show you is from the windpipe, the trachea. Finds the trachea. And the function, because of all the goblet cells and cilia, is a mucus escalator function. So it catches the debris, and the mucus is coughed up and out, and the cilia help beat it. Mucus escalator. Because essentially, this tissue in the trachea functions as a mucous membrane. On this micrograph, by the way, all of these are pictures that I took from our collection. And what I'll do, probably Friday, is I'll present all of our slides again, and I'll take new pictures for you that you can study. So I got these from when I taught it before. And the tissue you need to identify is up here. This is the trachea, and what you see beneath the epithelium is a submucosa. So from here on up is pretty much the epithelium on top of its basement membrane. And what I really like about this picture is it shows how this tissue is a mucous membrane, because these cuboidal cells are a gland. This is a multicellular gland, and you can see how the duct leads to the surface. Okay, usually you don't see that. All of these are glands, but you just can't see the duct leading to the surface because they didn't cut it in the exact right location. But this one they did, and I usually don't see this. So yeah, I got very excited. Hey, look at that. And even this right here, this is like, you know, pretend you shrink yourself and you're like inside and you're looking around. That's like you looking around. This is like an apical view of the of the pseudostratified columnar ET inside the gland here. It's pretty cool. Okay. Before I point to it, do you see the goblet cells? They're stuck in between. When I point to it. Now they're filled with mucus. They usually stay in light. See that there? Or that there? Yeah, those are goblet cells. Here's a closer up picture. Actually, that's not closer up. But this one has a more obvious goblet cell. That's a nice one. So what if I said identify, and I tuck this hairy stuff on top? What would you call it? That's the cilia, yeah. Here are the mucous glands down there. Okay. Here's a closer up picture. There's the goblet cell and the cilia, and this is definitely a pseudostratified columnar tissue. It's always pseudostratified columnar. Always. Can we move on? Questions?
And the goblet cells are always going to be in the top,
Yeah, they're always in the top.
Okay.
Yes. All right, this is a different kind of tissue, stratified squamous ET. Stratified squamous epithelial tissues are either keratinized or not. So some books like to say that they're not keratinized by saying non-keratinized, and you don't necessarily have to do that. If you don't see it there, just don't put keratin. Yeah, this is a good picture of it here. Now, because you have many layers on the apical surface, the locations are where you need protection against abrasion. And for example, you know, mouth, esophagus, vagina. Mouth, esophagus. So digestive tract, reproductive tract. Now, the function is, again, to protect against abrasion. And when you look at this tissue, again, only consider the apical cells. When you name it, because only the apical cells are stratified in squamous. The cells underneath are not. They're more like closer to cuboidal. So once again, the apical cell, just consider the top layers here. That makes it stratified squamous. This is part of the epithelium, but these cells are not squamous. They're young regenerative cells that will replace the top ones that get sloughed off. So when you slough off the top flat layers of cells, it's called desquamation. I don't know if I mentioned that before. Top layers get rubbed off and the bottom layers replaced. I think I might have mentioned that before. Anyways, here's a picture from our collection. There's no keratin. And again, just look at the top cells. And that's how you name it. So the whole epithelium is from here to here. But just these top layers make it a stratified squamous epithelium. Okay, so let's look at the keratinized stratified squamous ET. So I'm just going to tag on keratinized. So there's a nice picture of it. The stratum terms, I'll teach them Monday. Don't worry about that. In chapter 4, this is how you name this tissue. Stratified squamous epithelial tissue. It's keratinizing. So the location, skin. Specifically the epidermis, the top layer of skin. Same function, skin protects against abrasion, but because of the keratin, it kind of adds a waterproofing element. Epidermis is a word that means on top of the dermis. So on the bottom of the slide, you can see a different kind of tissue here. That would be the top part of the dermis. Okay. The keratin part on this slide is labeled stratum corneum, which is a word that means horn-shaped, because I guess they kind of look like horns, I guess. Well, anyways, many, many layers of keratin. Stratum corneum. So the keratinized part, stratum corneum. Essentially, what that layer is, the keratin, are just dead cells filled with keratin. That's it. In thick skin, you would have a very thick stratum corneum. On thin skin, that layer would be much thinner. So I'm going to write thick skin because that is considered thick. Thick skin, thick layer of keratin. You have thick skin, more so especially palms, sole of the feet. Thin skin, like the back of your leg, is a good example. Anyways, here's an unlabeled picture of our stratified, keratinizing stratified squamous epithelial tissue. I'll turn the lights off here again. And what you see is a stratified squamous epithelial tissue that is keratinizing. It starts down here and goes all the way to the top. But again, this part of it, from here to here, is stratum corneum, the dead keratin cells. This tiny layer right here is kind of where it's the stratified squamous part, okay? And we'll take a closer look at this in chapter 5 of integument. For now, just call this keratinizing stratified squamous epithelial tissue and move on. What I wanted to show you here, same slide, higher mag, it shows you the transition of where the cells begin to die. At the bottom layer of this epithelium, the cells are young and vibrant, and they're undergoing mitosis. As they get pushed to the top, where it starts to darken, that's where the cells die. So by the time you get to this layer, that's a clear layer, they're dead. Okay, they just continue to get pushed to the top until they get sloughed off in, disc formation. Oh yeah, callus is like a thickening of that, essentially. All right, so the transitional epithelium is the next tissue, and okay. So we name it differently because what we could see is it's a stratified tissue. Like I said before, histologists thought this tissue was the transition between stratified cuboidal and stratified columnar. So now we just call it transitional. But it's definitely a stratified epithelium. And when you look at the top cells, see how like kind of domey they are? I kind of drew a picture of this earlier. It's kind of what it looks like. Well, anyways, the locations, urinary tract. Now you haven't had that yet, but that includes like the urinary bladder, okay, which carries urine. The name of the structure that carries urine from kidney to bladder is the ureter. And the name of the tube that conveys urine to the outside world when you void your bladder is the urethra. So those are the major structures of the urinary tract. The function of this tissue is to stretch or distend. To accommodate the urine that's being transported. The hallmark of this tissue is on an apical cell, look for a binucleated cell on the top there. There's one there. Okay. Apical cell may be binucleated. All right, our picture. So this is from the ureter, I believe, and well, this big hill is the tissue, okay? And so you can't see the whole tube, but that's the lumen. And you just basically see all these stratified cells. It's transitional epithelium, that's simply what we call it. Okay, now I want to teach a different function of epithelial tissues. Epithelial tissue can be glandular. So I'm going to teach you about the different kinds of glands. I say epithelial tissue is glandular, and there's different kinds of glandular secretions, endocrine versus endocrine. So essentially, let's learn what those words mean. What is meant by endocrine, exocrine. Endocrine glands secrete hormones into the blood. Endocrine, exocrine is what I'm writing up here, and just kind of chart out the differences. All right, so first thing is you're a gland. What do you secrete? You're an epithelial cell. You have a glandular function. You're going to secrete something. So if it's an endocrine gland, secrete hormones into the blood. Exocrine function means you secrete something else. And as an example, I say mucus or serous fluids, and you don't secrete it into the bloodstream. You secrete it externally into something or onto something. So serous or mucus externally. It could be something else. There's a lot of things that have exocrine secretion. These are just a couple of examples. So these glands are more numerous. They require a duct in addition to the secretory cells. Endocrine glands are ductless. You don't need a duct, you just need access to a capillary. But these, because you're secreting it externally, you need a duct that leads to somewhere. All right, well, let's look at a picture of this. At the top, during development, the epithelium initially has a downgrowth during development. And if the gland has an endocrine function, the duct cells disintegrate and the glandular cells just have access to a blood vessel, whereas the duct cells will remain if the function is exocrine. Okay. All right, moving on. Epithelial tissue is glandular, but you could have unicellular or multicellular glands. Okay, so talking about the exocrine ones, it could be unicellular or multicellular. And basically, well, I think the mucus gland is a good example since we already looked at it. The unicellular mucus gland we already looked at was the goblet cell. That's the only one you got to know. The multicellular one that we saw was a seromucus gland. An example of that. So basically this picture is showing you the goblet cell in great detail. Our example of unicellular. It's one cell. Okay. For the goblet cells, since that's our prime example, remember where to look from. They're always on the top. They're always stuck in between other epithelial cells. They're a specialized cell that modifies the epithelium. There's a nice picture of it there. And now here's a table. This isn't like every physiology book. You'd be happy to know I don't hold you responsible for it. Just know that these are all multicellular glands. They all have ducts that lead to the surface. Yeah, I've been studying histology slides my whole career here. I've never seen this. kind of real tissues. I don't think you need to know it. It's never helped me. So don't worry about it. Just know that they're multicellular glands. There's different kinds. Here's the one you got to know. The one I already showed you a picture of. One of the easier questions I'll ask you. Is this gland unicellular or multicellular? You see many cells, it's multicellular, right? And if I point to the single goblet cell, that's the only thing you got to know. That's a good example of a unicellular gland that we see in A and P. Okay, so the functions of these glands is secretion. There are different kinds of secretion. A secretion is it oozes out. So how does the secretion get out of the cell? There's merocrine and holocrine. What the picture is showing you is that there are secretory vesicles that are fusing with the apical surface to get the secretion out. So the mechanism is by exocytosis. That's what we call that. Secretion by exocytosis. Now the other one, polosecretion, the apical layer of cells is basically all ruptured. So the ruptured cell is how the secretory product gets released. That's holocrine secretion. Apical layer of cells rupture. Okay, so that's glandular function. We're going to shift topics here. Now that I've taught all the epithelial tissues and all their functions, one major function of epithelial tissues is they are a component of membranes. So in A and P, when we say membrane, it has a very specific meaning. Membrane is an epithelial tissue overlying the basement membrane, the connective tissue basement membrane. So it's the ET overlying the connective tissue basement membrane. Those two things is a membrane. And they could be dry, mucus, serous. Here's an example of a cutaneous membrane, skin. You know, I love how like the books, they like to call the same thing different things. We already talked about skin. We called it a keratinizing, stratified, squamous epithelial tissue. That's how you would name the tissue. It's skin. It's also considered a dry membrane. When we talk about it in chapter 5, the chapter on skin, we'll call it the epidermis. So it's like, 4 different ways to call the same thing, skin. So know that it's a cutaneous membrane. It's dry. And it's skin. The keratinizing, stratified, squamous ET. Right there. We already looked at that. Okay, so mucous membranes, they line body cavities and they're open to the exterior. A good example is what we've been talking about, the airways, okay? Trachea, mucous membrane. You'll notice that they like to put a O-U-S for mucus. I tend to leave that O out. Both spellings of mucus are acceptable. So mucous membrane, well, they're not dry, they're wet because they have mucus. It goes without saying, it's mucus, and you know mucus isn't dry. A good example of that is in the trachea. I'll just list that as the example because I've been showing you slides of that all morning. Okay, here it is again. Same slide again. It's a great example of a mucous membrane. Okay, this tissue. I mean, in naming the tissue, it was a ciliated, pseudostratified columnar epithelial tissue with goblet cells. It's a mucous membrane, right? It's from the trachea. All right, this is a serous membrane. Show me where they are. Serous membranes are also wet. There's a tiny layer of wet secretion. It's not mucus, it's called a transduit. That's the name of the wet secretion. And serous membranes, well, for example, the trachea was a pseudostratified columnar. Serous membranes are usually a simple squamous epithelial tissue. Okay, that's the kind of epithelium we usually see in membranes. And to be thorough, let me just kind of list the type of epithelia we see in the trachea. That was a pseudostratified columnar. Okay, that's the slide I just showed you. And for the skin, I had said it was a stratified squamous ET. So we looked at all this, we're just calling them what their function is. And the serous membranes, they line body cavities, they surround major organs. If it lines the body cavity, if you look at the picture, where it says peritoneum, that's the lining of the body cavity, okay? So location is peritoneum. This serous membrane also surrounds the heart and lungs. If it surrounds the heart, it's called the pericardium. If it surrounds the lungs, it's called the pleura. So pericardium, as well as pleura. There's an expression called the 3 P's of anatomy, and it's peritoneum, pericardium, pleura. Those are the 3 P's. Helps you remember where all the serous membranes are. Well, anyways, we looked at mesothelium. That is the serous membrane that lines the peritoneum. We looked at this before. Okay, and that's what the tissue appears like under the microscope. Again, that's mesothelium. Heard of mesothelioma? Well, that's a cancer of the pleura. So I've heard that in advanced cases, they have to remove the whole lung. So it's very serious. Okay, well, anyways, that's the end of the slides, actually. And these slides, I say histology practice, I don't teach them anymore. Because ever since we got our scope camera, I show you our slides. But they're still useful pictures. I thought I'd leave them here if you want to practice yourself. Go right ahead. There's still fun pictures to look at if you're studying. But I'll present our slides later on, probably Friday. Today what I'm going to do is I'm going to continue on with connective tissues. Done with epithelium. I'm opening the connective tissue slides. So this batch of slides have connective tissue and muscle tissue. I don't even get to muscle today, but we learned how we classify the epithelial tissues. Connective tissues are a very different category. in epithelial tissue, we kind of look at how the cells are arranged. But in connective tissues, you barely notice the cells. Mostly what you see is the extracellular matrix and fibers. That's really the thing to pay attention to. So this is the second of 4 basic tissue types. The other 2, well, the 2 I taught so far, I taught epithelial. I'm going to teach connective, going to teach muscle. Nervous tissue, I'll save for the nervous tissue chapter. So we'll really do like 3 of the 4 basic tissue types. But there's a lot of connective tissues to go through. And for connective tissues, focus on the extracellular matrix. Now, the extracellular matrix is a ground substance and fibers. Ground substance, there's a lot of water content, there's a lot of proteins in it. I have that term there called gags and water. Gags are like these bottle brush proteins. They create osmotic pressure to help suck fluids into the tissue. Okay, so I'll put that there to remind myself it's part of the ground substance. Really, what you need to focus on are all the different kinds of fibers. Fibers, and being able to identify them will help you identify the tissue. That's really your clue, in my opinion. You know, what you're looking at. And I put the 3 different fiber types we commonly see: collagen, elastic, reticular fibers. Collagen, elastic, reticular fibers are the commonest. In terms of cells that you see, leukocytes, white blood cells. This slide has like macrophage, lymphocyte, mast cell, neutrophil. Those are all kinds of leukocytes. Okay. And of course, what we mostly see are the cells that secrete the fibers. Okay. And those are fibrocytes or fibroblasts. And you usually see a lot of adipocytes, commonly known as fat cells. And those are mostly for nutrition. Well, anyways, study this graph to learn how the connective tissues are classified. Okay, so let's kind of go through the different classes here. That top category is probably the most extensive subclass of connective tissues, and they just call it connective tissue proper. Okay? And all the connective tissues, it's kind of hard to like say what their functions are because there's so many different kinds. So I'll just kind of tell you the function as we go through each type. Anyways, just to classify them, the subcategory of connective tissue proper, it's based on how the fibers pack together. If the fibers are more or less loosely packed, it's like a loose CT. CT being connective tissue. If they're densely packed, you can call it a dense CT. but then there's subcategories of each loose and dense, okay? So for the loose ones, I'm right here, these three, areolar, adipose, and reticular, those are the ones you gotta know. Areolar, adipose, reticular. We'll look at pictures of all of them. Loosely packed fibers, and you should be able to identify them. For the dense ones, there's dense, regular, dense, irregular. If the fiber is packed in a way, and they're all oriented more or less in the same direction, say it like this, aligned the same way, that would be like a dense, regular connective tissue. But if they're oriented any which way, there's no regular orientation, that would be dense, irregular connective tissue. So the orientation of the fibers is important. Because if all the fibers are oriented the same way as in dense regular, and you pull in that same orientation, you're very good at resisting that pulling force. They call it tension. Tension is a pulling force in physics. So dense regular connective tissues are very good at resisting tension. Resist tension. A good example of that is our tendons and ligaments, right, that move your joints and you contract your muscles. Tendons, ligaments. We'll look at pictures of those. Basically, you can resist tension. However, if you exerted a force, say that was perpendicular to the orientation, that would not resist at all. It would shear quite easily. Okay, so shearing is a tearing force. So you can resist tension, but you could shear easily. You can't resist shearing forces. However, if you're the dense irregular kind, you could pull this in any direction and resist the shearing because the fibers are in any direction you could possibly pull. So we say dense irregular connective tissues resist shearing forces. The last category of the dense tissues are elastic tissues. Now, elastic tissues are made to stretch. Okay, they're categorized as dense. Usually, when I look for them on slides, I look for the blackest staining fibers. They stain black. They're very dark. Black. In terms of the dye that they pick up. You got other categories there. You have cartilage, bone, and blood as other subcategories. Cartilage, the function of cartilage, is part of the skeletal system, and well, cartilage is basically bone, just without the calcium salts. And cartilage has a high collagen content. Okay. Lots of collagen. Let's see here. The type of cartilage that has a lot of collagen is this first one here called a hyaline cartilage. So collagen fibers are the type of fiber you would see in hyaline cartilage. I'll put hyaline in parentheses. They have elastic cartilage listed, so you'd see a lot of elastic fibers in the, oh, obviously, you'd see a lot of elastic fibers in elastic cartilage. The fibrous nature of fibrocartilage that one's very unique. And the example I want you to know is in the intervertebral disc. So for these other ones, I'll give you examples when I get to them. But this one, we're only going to look at one kind, the kind of cartilage in the intervertebral disc. Let me pause a second. I just got to change my battery. I'll be right back. All right, we're good. For cartilage, when we look at the pictures, the one thing I want you to look for is the cell type. Look for the chondrocyte. The prefix chondro means cartilage. Cyte means cell, obviously, and they have a little space that surrounds them. The space that the chondrocytes occupy is called the lacuna. So we always say chondrocytes and lacuna. That's the cell type there. All right, the other two tissue types is bone and blood. Bone, also called osseous tissue. Ossification is simply cartilage that takes up calcium and makes the salts, and bone is cartilage that has been ossified. Okay, we'll learn that process in another chapter. But I guess I should at least put compact bone. That's the one I'll show you today, compact bone. There's also spongy bone, but we'll just look at the compact bone today. We have blood. We'll go over here. Whole blood is actually about 55% fluid or plasma. In a drop of blood on a slide, you really can't see that. The rest of blood are the what are called the formed elements. The blood cells, which include erythrocytes, leukocytes, or white blood cells, and thrombocytes. So you really notice the blood cells, not the plasma, in a drop of blood, and we'll take a look at that. Okay, well, anyways, those are the categories of connective tissue. Let's do the connective tissue proper category first, starting with the loose ones. And here are the ones you got to know. We'll go through them one at a time. There's the areola. Now, areolar tissue, I would note three things: the collagen fibers, the elastic fibers, and the fibroblast cells. Those are the three things I would have you identify in this tissue, in addition to just identifying the tissue, right? So note the collagen fibers. Now the picture is pointing to it. I mean, what color would you use to describe the collagen fibers? Pink, right? That beautiful pink color. Now what color would you use to describe those hair-like elastic fibers? Like more of a dark purple, okay? So maybe the colors can be your clue, as well as the thickness of the fibers. The collagen fibers are much more thick. The elastic fibers are very thin and stringy in this tissue. They're not always like that, but they are here. Collagen fibers, pink, dark and thin. So that's how I would describe them. Note the fibroblasts. In terms of location, they give you one example, like the basement membrane. They're all over the place, okay? I'll give you one here. Location, basement membrane. Underlying the underlying connective tissue of epithelia. Okay. Here's a picture I took from our collection. It's not as pretty. The colors aren't as vibrant. I think you should still be able to identify everything. So what you see here, if I point to a dark circular object like this or this or this, what do you think I'm going for? The dark circular structure would be considered the fibroblast. What if I point to the dark hair-like thing? The elastic ones. If I try to point to a pink thing, then I'm going for the collagen. Okay? This is areolar tissue. Areolar tissue. It's a good one to start with. Now we have adipose. Adipose, there are basically no fibers that I can see. Mostly what I see in this tissue are the adipocytes. Whoops, let me write adipose, then the cell is adipocytes. Tiny bubbles, and well, the cell is filled with cholesterol, which does not take a stain, so they're difficult to view. You can barely make out their cell membrane there. So it's like you have a cell, and the nucleus is pushed to the outer edge, and the adipocyte is filled with cholesterol that doesn't take up a stain. Don't see much else. Here's a picture I took from our collection of the adipocytes. That's all you see. You see the tiny bubbles, it's adipose. Okay. Now adipose, most notably, is wherever your body has fat. In, for example, subcutaneous layer, skin deep.
Are the bubbles the little black dots?
The bubbles are the adipocytes.
Okay, okay.
Yes. Subcutaneous tissue. So the subcutaneous tissue is the location where you have most of your adipose. Body fat, on average, 20% to 30%. And then the subcutaneous layer, I guess one function is it cushions, it's for warmth. Although, in most places you just put on a jacket, okay? But if you're stranded in the forest overnight, this time of the year, how much body fat you have could save your life, okay? So it's something you talk about more in other classes. Like I remember when I was an undergrad, I had to answer this really hard question. The situation was you're somehow you get stranded at sea, and you're a mile from shore. And I was asked, should I swim back or should I just wait there to be rescued? And I had to answer the question based upon before hypothermia sets in. And part of the equation was I had to figure out how many millimeters of fat were there. So it does become important in survival situations, but we don't need to worry about that here. Just identify adipose. All right, this loose CT is a reticular tissue. Look for those reticular fibers. They're very branchy and black looking. Reticular fibers. Reticular in anatomy means complex network. So the reticular fibers, they form a complex network. Reticular fibers form a complex network for cells to reside. The function of this tissue, it's like a mesh to filter things. Tissue forms a mesh to filter whatever. I mean, it depends. The commonest places, see, they have a picture of spleen. All your filter organs, like spleen, it's a good example. Locations, spleen. I think the lymph node's the best example. Well, you haven't had the chapter on those organs yet, but basically, the spleen filters the blood. The lymph node filters lymph. Lymph is a fluid. Okay. So one filters blood, one filters lymph. Obviously, the lymph node filters lymph. So pretend it's the lymph node. The lymph node is packed out with lymphocytes, with all this reticular tissue, and you're straining all of the body fluids through that structure. If there's a pathogen, you slow the flow of the fluids down through this mesh, and if there's a pathogen there, the lymphocytes that occupy the lymph node might be able to find it to mount an immune response. So that's basically the function. Lymph is fluid. So your blood when you get to the capillaries, some of the plasma filters out. That filtered plasma that's in the tissue is now lymph, and the lymphatic system will pick it up so you don't swell. Swelling in your extremities, edema, is basically a failure of the lymph nodes, or excuse me, the lymph vessels. And if you walk around and improve your circulation, you'll prevent the swelling. Okay. Well, anyways, I think this picture kind of more shows how this is like a mesh, a sieve. So all the pinkish things you see, this is the lower bag, are the cells of the organ. All the blackish structures, if I had you identify it, what do you think I'm going for? Well, there's only one fiber type. What is it? Reticular. And if you can remember that, you'll know that it's a reticular tissue. Functions like a mesh net. So move on. Okay, moving on. This is dense, regular connective tissue. Dense, regular connective tissue. Regular CT. So if you look at the picture, I just see a bunch of collagen fibers packed tightly. In the same orientation, which makes it the regular kind. That's all you see. Maybe you see a couple of squashed nuclei of the fibroblasts, so I guess I can have you identify that. Fibroblast. So tendon, ligament are the best examples. I believe this is from tendon. This is a 430 magnification. Okay, remember, we only go to 400, right? So location, tendon, but ligament is also a good example. Well, in case you didn't know, a tendon is a dense, regular connective tissue that connects the muscle to the bone, so you can move joints. So tendons connect muscle to bone. And like in a joint, a ligament connects a bone to another bone in a joint. So it connects bone to bone. Here's our dense, regular connective tissue. It looks like plywood, kind of. Well, stare at it for a little bit. See if you can notice the orientation of the collagen fibers. They're kind of like all running this way. What if I point to, I don't know, that thing, that dark thing? What do you think I'm going for? Fibroblast. That's all there is. Fibroblast and all the other stuff is collagen. That's tendon, by the way. Okay, here's dense irregular. I don't like the picture from the book. Let's go with this one here. Actually, let's go with this one here. The next tissue type is dense irregular connective tissue. So dense irregular connective tissue, well, you can look at the picture. There's no regular orientation to the collagen fibers. The collagen bundles are oriented every which way, okay? It's irregular. So I think that's an apt description. The example, you gotta know dermis of skin. Here's a closer picture. When you cut this tissue, all the fibers are just going every which way. It just all looks all mixed up. So go back a couple of slides to show you an illustration of where the dermis is. It's like putting meat through the meat grinder. And all the collagen fiber bundles are all mixed up. And well, here's a thick layer of skin. There's actually 3 layers here on the picture on the left. Up top, you've got the epidermis. The dermis is from here to here, okay? And then you have the subcutaneous tissue, which is full of fat. So the dermis is what I want you to identify as an example of dense, irregular connective tissue. All right, so moving on to dense tissue, elastic tissue. The elastic tissue, the elastic fibers, they're very dark, they're very black. They stay black. And okay, let me turn off the slides. The example, I'm sorry, I'll turn off the lights, but the example here is the vessel wall. That's aorta, actually. That's a large artery. It's a good location. Large arteries, I mean the biggest one being aorta. The function of elastic tissue in a blood vessel is to accommodate the cardiac output. Every time your heart pumps blood, the vessel has to stretch. And then there's an elastic recoil when the heart's relaxed to push blood forward. So it's good to have a high elastic content. In fact, the largest category of arteries are called elastic arteries. Elastic arteries have what's called, quote unquote, elastic recoil while the heart is in diastole. That's the term, elastic recoil. While the heart is in diastole. Diastole is the phase of the cardiac cycle when the heart muscle is relaxed. So when the heart is relaxed, it's not providing force to pump blood, right? So it's very important that this elastic recoil occurs so blood can keep moving forward during the time the heart is relaxed. So that's a very important concept you'll relearn in 431 when we talk about cardio. But for here, I'm just giving you a physiology reason for having elastic waistband in your blood vessels. Okay, let me turn the lights off. So the elastic fibers, if you look at that thick layer of vessel wall, I see 2 colors, black and pink. So what color are the elastic fibers?
Black.
The black. The pink is actually smooth muscle. But you know, we're kind of interested in the elastic tissue now. Now here's a picture I took of a large artery. Okay. This outer layer has no elastic fibers, but this inner layer is called the tunica media. It has a lot of elastic fibers. But wherever you see the blackish tissue is elastic tissue within the vessel wall. Here's a closer up picture of it, so you can kind of make out the individual elastic fibers there. Okay, it's considered a dense connective tissue. this is a good place to stop. We'll pick it up here Friday? Yeah. Lose track of my days of the weekend. See you Friday.